Prasanna. I am working as a biology teacher in Montessori English Medium School, Ellarpeta, Palpur. Today we are going to discuss about nutrition lesson. This is a food supplying system. Now we have to know about first of all nutrition. Nutrition is in taking food and consuming it and then utilizing the food. It is called as nutrition. Nutrition basically nothing but utilization of nutrients. Utilization of nutrients. Nutrients are two types. These are macronutrients and micronutrients. Based upon their need, macronutrients are needed in huge amount. These are called as macronutrients. The nutrients which are needed in minute quantity, those are called as micronutrients. Examples for the macronutrients are the carbohydrates, fats, proteins. Examples for the micronutrients are the vitamins and minerals. Okay. Based upon the type of nutrition, the organisms are categorized into two types. Those are autotrophs and then heterotrophs. Okay, auto means cell. Trophs means feeding. From this meaning, the definition for autotrophs, we can say that the organism can synthesize their own food material by its own by using some inorganic materials through a process called as photosynthesis. Through a process called as photosynthesis. Okay, going to the heterotrophs. Hetero means others. Trophs means feeding. That means heterotrophs not able to prepare their own food. Okay, they can depend upon others for getting food materials. These heterotrophs are categorized into three types. Those are holojoint type of nutrition, dog, etc. animals. Okay, these are doing their nutrition by using holozoic type of nutrition. That means nothing but ingestion, digestion, utilization and then excretion. This type of nutrition is called as holozoic nutrition. Then parasitic nutrition. Then parasitic nutrition. In this case, the organism can take shelter, can take food, can take all their needs from other organisms are called as parasitic nutrition. That means especially microorganisms like bacteria, viruses and some microarthropods having this type of parasitic mode of nutrition. That means they can take shelter. For example, consider me or consider any human beings. The skin of any human being having some crores of microorganisms like bacteria and viruses, but they can't able to cause any disease. Okay, against our resistance system present in the body. Okay, that type of nutrition is called as parasitic nutrition. Then here I mentioned take shelter from host. Take shelter from host. What is meant by host here? What is meant by host here? The organism which can give shelter, which can give food and which can give other needs wanted by the parasitic organism is called as host. Okay, is it clear? Okay, for example, me is a host for so many types of microorganisms. Okay, then saprophytic. Then saprophytic. The mode of nutrition is taking food from dead and decaying matter. Taking food from dead and decaying matter. Dead and decaying matter. Okay? That means examples for this saprophytic nutrition, the organisms like fungi includes molds, yeast, okay, mushrooms. Mushrooms are included under this category saprophytic nutrition. Nothing but the getting of nutrients from dead and decaying matter. It is called as saprophytic nutrition. It is overall view of nutrition and types of nutrition. Now, I am going to explain about experiments to prove the starch is released from the starch is stored in the plant after completion of photosynthesis. Okay, first of all, I want to explain about starch identification test. Starch identification test. Okay, first of all, you have to select one potted plant. First of all, you have to select one potted plant and keep it in dark room up to 3 to 4 days or 1 week. Why? Because to keep that plant in sunlight up to 3 to 4 hours. Up to 3 to 4 hours. Okay? Okay, after 3 to 4 hours, photosynthesis was completed. Then you have to pluck. Is it your own choice? You have to pluck a leaf. You have to pluck a leaf and keep this selected leaf in a test tube. In a test tube having alcohol, methylated spirit or ethanol. Okay? In the, can I repeat it again? Okay. Keep this leaf in the test tube and pour ethanol or methylated spirit in the test tube and keep this test tube having methylated spirit having leaf in a water beaker. There is no water, but just assume it. Okay. Okay. Keep it in water beaker. Then this water beaker is allowed to heat or boil under flame. Okay. You have to create a flame here. Then flame. After boiling of water, this boiling water can also boil the alcohol in the test tube also. After boiling, this alcohol bleaches the chlorophyll present in that leaf. Okay, this is a green colored pigment having leaf. Due to boiling, the alcohol bleaches the chlorophyll pigment in that leaf. So, after dechlorophyllation, you have to keep carefully. You have to take carefully that leaf and keep it in a watch glass or Petri dish. Okay, I am here taking only watch glass here. Okay, keep it on a watch glass. Then add 3 to 4 drops. 3 to 4 drops of iodine solution. Okay, after addition of iodine solution, the leaf and for example, after bleaching of chlorophyll, the leaf will convert into white pale color. Okay, after addition of iodine drops, this pale white color will convert into dark blue. Or black color. This indicates that starch is formed after completion of photosynthesis. Okay? Am I clear to all? Okay. Then I am going to explain about CO2. CO2 is essential for photosynthesis. CO2 is essential for photosynthesis. Okay. Take a potted plant and do as I said in the before experiment. That means keep it in a dark room, etc. Okay. Then take a selected leaf which is in contact with the plant. No need to pluck the leaf. Which is in contact with the plant body. Okay, am I clear? Which is in contact with the plant body. Now you have to take a wide mouthed this. You have to keep this leaf carefully into splitted region of the core. Then you have to cover like this. Then you have to cover like this. This is a leaf and this is core. Okay. To prevent extra CO2 enters into bottle from the atmosphere, you have to protect glass bottle with a grease covering. Okay, you have to cover it with a grease. Okay, na? Okay, grease. Okay, or Vaseline, anything, etc. Okay, then there is no way to enter the CO2 into the bottle. But there is some CO2 present in the bottle. So the CO2 is consumed by the KOH. That is the need of KOH is to consume the CO2 already present in the bottle. Okay, it can absorb the CO2 into itself. Okay, so there is no CO2. There is no CO2 for the part that is left in the bottle. Part of leaf that is left in the bottle. But one thing, CO2 is available for the part of leaf that is present just above the bottle surface. Okay, CO2 is present for above the surface, not available for the leaf part present inside the bottle. Okay, after 3 to 4 hours, it is exposed to sunlight. After 3 to 4 hours, photosynthesis process is completed. Then you have to pluck this leaf and do as the starch experiment. And for example, take alcohol in this test tube and then boil it in a water pot. After that, chlorophyll will be bleached out. Then take this leaf in a petri dish. The region of leaf which is present outside the bottle. That means it is exposed to sunlight. It is exposed to CO2 as well. So it can convert into blue or black color by using iodine test. Okay, here I am doing only iodine test. By adding 2 to 3 drops of iodine solution, the leaf which is present outside the bottle converts into blue or black color. But the leaf present inside the bottle, it is not exposed to CO2, but it is exposed to sunlight. It is exposed to sunlight, but there is no exposure to the CO2. Why? Because here I am applied grease to prevent the entry of the CO2. Okay, there is no CO2 here. That's why you cannot change its color. No change in color. No change in color. From this, I conclude that CO2 is essential for the photosynthesis. By using CO2 only, this leaf part can synthesize the starch. Okay, after that I did iodine test, then only it will give blue or black color. Okay? Okay, next I am going to explain about sunlight. Sunlight is essential for the photosynthesis. Sunlight is essential for the photosynthesis. Okay. Now you have to take again always you take a potted plant and keep it which is exposed to the sunlight, the region which is exposed to the sunlight will convert into blue or black color. Will convert into blue or black color. Okay. Here, cutted design also converts into blue or black color. Why? Because there is place to enter the sunlight into the leaf. But in the area where the black pepper cover does not allow the sunlight. Okay. So, from this experiment we conclude that where the region which is exposed to the sunlight is converted into blue or black color, we can conclude that sunlight is essential for the photosynthesis. Thank you.